Experimental Operation Demonstration of RAA Nucleic Acid Amplification Kit. Fluorescent method. What you need. Nucleic acid template. Forward primer. Reverse primer. Probe. RAA Nucleic Acid Amplification Kit Fluorescent method. Mini centrifuge. Isothermal oscillation mixer. Isothermal nucleic acid amplification detector. Centrifuge tube rack. PCR tube rack, pipette tips. Kit provides purified water, buffer, magnesium acetate, reaction unit, forward primer of positive control, reverse primer of positive control, probe of positive control, instruction manual. Before you begin the experiment, you will first turn on B6100 and F1620 for preheating. Open the software on the desktop. Select the serial port number corresponding to the instrument and connect the communication. The mixture was prepared according to the reaction system specified in the manual. The purified water, buffer, forward primer, reverse primer, and probe were added to the 1.5 milliliter centrifuge tube in sequence. It is recommended to prepare X plus 2 tubes of mixture. X is equal to the number of nucleic acid samples. The above mixture was mixed by hand and centrifuged briefly. Take out X plus 1 tubes of reaction units and mark on the tube lid. Open the lid of the reaction tube and add the prepared mixture to the reaction tube. Close the lid of the reaction tube. Flick each reaction tube with hand and collect them by mini centrifuge. This step cannot be finished by vortex oscillator. Transfer the reaction tubes to sample addition area. Open the lid of each reaction tube. Add 2.5 microliters magnesium acetate in the tube lid. Add nucleic acid template to the reaction tube. The purified water can be used as negative control. When adding samples, it is recommended to add samples according to the sample concentration from low to high. To avoid cross-contamination, please make sure that the lid is closed tightly immediately after one sample is added. Symmetrically place the reaction tubes into the RAA B6108. Click the Pre-treatment button to mix and centrifuge. Place the tubes into RAA F1620. Click the Run button to conduct fluorescence detection. Real-time amplification curve as shown in the video. Amplification results can be observed in 5 to 20 minutes. After the end of the run, set the slope parameter at 20. Check the slope box, and the software will display the analysis result on the left side. Notes: after the completion of the experiment, the opening of the lid is forbidden to avoid the aerosol pollution caused by amplification products. It is recommended to add samples in a special edition area or biosafety cabinet to avoid reagent contamination caused by positive DNA. In this kit, the forward primer of positive control, reverse primer of positive control, and probe of positive control can only be used to amplify the supporting positive quality control. If the user needs to amplify other nucleic acid sequence, the primer and probe need to be designed by his own. Please contact the manufacturer to obtain the primer and probe design principle.